So structure and shape determines function. This is an important principle of molecules and chemistry and biology, biochemistry. We need to know the shape of a molecule in order to know what it does. So here is a nucleotide. This is the building block of DNA. They have a nitrogenous base here. Here's a pentose sugar or a ribose sugar, and then a phosphate group. These three components build a nucleotide. The phosphodiester bond is determined by the phosphate group and the ribose sugar. If this is a double-stranded DNA molecule, or as you'll find out, that it forms into a double helix. The red part is the sugar phosphate backbone, and in the middle, pairing up, we have the nitrogenous bases. We'll talk about those in a little while. And that's basically the main structure of DNA. Now, how do we know if it's DNA or if it's RNA? I'm sure you've heard of RNA. Well, it's all determined by the sugar molecule. Again, here is our nucleotide. First, we need to count the carbons, figure out what the numbers of the carbons are. So here is our anomeric carbon. As you know from Bio 180, it's between the two oxygen molecules, and then we label them 1 through 5. Pentose sugar means there are 5 carbons. Now, off of the 2' carbon here, if a hydroxyl group is present, then we know it's RNA. If it's just a hydrogen, if that oxygen is missing, then it's DNA. RNA means ribose nucleic acid. DNA means deoxyribose nucleic acid, so it's deoxygenated, that oxygen is missing, DNA. If here's our DNA strand, we've got a 5' end and a 3' end. This is known as directionality. If one end here, we draw this nucleotide, number all the carbons, you'll see that the 5' end is sticking out in that direction, and if this nucleotide was on the other end of the strand, then the 3' end would be showing there. And then this is how they pair up. We have the phosphate and sugar molecules pairing up to make phosphodiester bonds right there. Between the 5' and 3' carbon, so it's known as a 5', 3' phosphodiester bond. So here's a DNA double helix. Now let's talk about base pairing. This is really important for the function of DNA. So nitrogenous bases do the pairing. We have 2 types of nitrogenous bases, purines and pyrimidines. The pyrimidines are guanine and adenine. The pyrimidines are cytosine, uracil, and thymine. GA, guanine and adenine. How I remember this is God and his angels are pure. That makes it easy for me. And purines are a 2-ring structure. Pyrimidines are a 1-ring structure though. And as you see, CUT, cut. I remember this by saying cut the purine in half, or cut the 2-ring into 1 ring. Now, all of these are present in DNA except uracil. Uracil replaces thymine in RNA. Don't forget that, that's really important. Now, purines always paired to pyrimidines in double-stranded DNA. And RNA, I guess, yes. The purines always pair with pyrimidines. This is important. G bonds with C, guanine to cytosine with a triple hydrogen bond. This is important when replication and transcription comes into play. Adenine and thymine always pair together with a double hydrogen bond, and adenine pairs with uracil in RNA. Now, a scientist named Chargaff discovered this and determined the rule that purines, the ratio of purines to pyrimidines will always be 1-to-1 in double-stranded DNA and RNA. So DNA structure on the large scale, this is an important thing for you to understand. Eukaryotes and prokaryotes are slightly different. Eukaryotes have linear DNA, and the chromosomes look like this and are stored in the nucleus. Prokaryotes have circular DNA, kind of looks like that, I guess, and they're stored in the nucleoid. Forgive my really bad drawing and might be even inaccurate, I'm sorry. Prokaryotic DNA is packaged just via supercoiling. It kind of wrap up and just form into the nucleoid there in the middle of the bacteria cell. Eukaryotic DNA, however, is packaged using histones. So if here's our double-stranded DNA, remember there's like 6 feet of DNA in one nucleus that has to be wrapped up and really tightly packaged, and so wrapped around histones, these little greeny yellow circles are the histone proteins. The DNA wraps around that, and it keeps doing so for a few histones, I'm not sure the exact number, and they form into what's called a nucleosome, and it keeps on going from there, but I won't draw it, I'll just talk you through it. So the DNA double helix wraps around the histone proteins, which form into a nucleosome, which further coil into chromatin fiber. The chromatin fiber then undergoes more coiling and eventually is formed into a chromosome. And this is how DNA is tightly packaged in the nucleus of eukaryotic cells. And last of all, something I forgot to mention earlier is a principle of DNA called antiparallel. I think I spelled that wrong, oh well. So I drew a DNA strand, 5', 3'. This end, as we know, has got the 5' part of the nucleotide sticking out. And of course there are thousands, if not millions, who knows how many nucleotides along this strand. And on this end, the nucleotide, the 3' end is sticking out. Well, in double-stranded DNA and RNA, it's antiparallel, meaning the opposite strand runs in the opposite direction. So this side would be 3' and this side would be 5'. So one strand is going in this direction and then the other strand is going in that direction. So that's something called the antiparallel. The DNA or the RNA in double strand form is antiparallel to each other. Make sure that you understand that and recognize that when you are looking at DNA molecules. I hope this video helped. Thank you for watching.